All right. Hello, everybody who is watching and anybody who's watching later, hopefully. This is the episode was originally titled, you know, Evolution, Enzymes, Extremophiles, Protein Synthesis and Protein Folding, and the Future of Science and Technology with Austin Sims and T Savo, but that might warrant changing later. It seems T is busy, or he's, as often happened, somebody, possibly me, or someone else misunderstood the time zones and converted them poorly. Haven't heard back from T, but hopefully he messages me back or jumps into the room at some point. I've got all the means of communication open, just waiting to hear back from him. If he does join us, then we'll pivot back to the original topic and talk about that. But for now, it's just going to be me and Mike instead, talking about some things that I'm hoping to get a better handle on here.
Me too.
So let's talk about him again. We've talked about him a few times before, but I don't think we've specifically this is a good chance for me too, because I have my computer set up. We can go on and do some actual comparisons, and I can learn how to do this a little bit live. So
Okay.
you said, so we're going to talk about first how Mike has been shoveling and sifting through genetic data by hand as just an interesting Samaritan, and then you had some stuff you wanted to. We're going to go over chromosome 2 a little bit more, because there is a lot to go over there, and we've only gone over half of it, 1/14 of it, something like that.
Yeah, 1/14.
Yeah.
I'm way down the rabbit hole on this stuff, and I actually get sick from doing this work, but I love it. I love doing it, but it actually freaks me the hell out sometimes. So anyway,
I feel that way about science all the time, Mike. That's totally
yeah. Yep. Yeah, sometimes I feel like I'm losing my moorings,
Yeah.
like my brain is going to fly off, like a flywheel. Just all of a sudden, the bearings go to hell and it flies out.
Yeah. This is something that's common with people who do scientific research and get to high levels. That feeling of unmooring is fairly common.
Yeah.
You're losing touch with reality, because you're learning more and more how reality breaks down, and how there's all these exceptions, and you'll never have a wide enough sample pool, and on and on.
Yep. So what I did is I downloaded the entire I downloaded the whole genome, every single chromosome. You get it in one big batch. And then I used something called awk, which is a Linux command, to break it up into each chromosome. So I have chromosome 2 for the human from the latest, greatest build. And then I got chromosome 2A and 2B from the chimp, right? And this is kind of a diagram right here that shows you where the fusion site is. The fusion site is that orange part right there.
Yeah.
So I have this entire chromosome 2A, and I went and I did some work and found out that gene does not, there's no evidence for a gene that crosses the fusion site. There's one exon that's sometimes translated on the left, on the upper part of the fusion site, but it very seldom gets translated. And when I did a blast search on it, it turns out it's not related to the part that does get translated. So in the latest annotations, they're showing it not across the fusion site. So there goes one of their big talking points right there.
Okay, so we immediately hit the thing where I'm at. You're so far beyond my understanding on this, because I'm still just struggling with how to use things like BLAST itself. So that's accessible on what? NCBI? You said you use US.
Yep. NCBI and US UCSC, University of California, Santa Clara, or Scrotum, whichever you choose. And it's their genome browser. So if you search for UCSC genome browser, you'll come up with this page right here, right?
Okay.
And this allows you to go out and do things like you can pick a genome up here. I went to the upper, I'm going to make that a little bit lower so you can see what we got here. Let's see. Okay.
I just pulled it up. I'm looking at Genome Browser, BLAT, in silico PCR. Ooh, talking about the polymerase chain reaction on here. That thing is so fascinating to me. Okay, this is really cool.
Okay, do you see the same screen that I see? Okay.
No. What did you click on, like genomes or genome browser or something? I see the links up here.
Okay. UCSC genome browser. I'll go ahead and put it in. I'm going to put it in public chat so people can follow along if they want.
Yeah, that's a good idea.
Okay. And then you can click on that. So that's it.
Okay. I clicked on something, and right now I am on the human genome.
Oh, okay.
Wow.
Are you looking at UCSC?
UCSC genome browser on human GRCH38/HG38. So I'm guessing these are two genes.
Okay, go back up here to in the upper left. I think if you click on that's Santa Cruz. Shit, that's going to give me. Let's see, genome browser UCSC. If you get to this page, you need to get to this page.
I was on that page previously. Okay. Genome. Okay. Here we go. Here, I'm back on that page now.
Cool. Get back there. Okay. Now in the upper left, there's genome browser. You can go right to back to genome browser, and it's going to go to whatever you were last working on.
Yes. Okay. So I've probably used this before, and you might have accidentally. Okay. Well, good to know. I was better at this than I thought I was. Okay. Cool.
Yeah. So if you look up here at genomes, you get a drop-down list, and I could go directly to it, but I want to show you something else.
Okay. Yes. So all right.
Just click on genomes.
One of the genomes I was looking at previously is one of the main ones here. It was the top one, the GRCH38HD38.
Yeah, that's the one that's most annotated, but then there's a newer one called HS something or another. But if you go down here,
that would be the T2T CHM13HS1. The T2T being telomere to telomere. Yeah. Okay.
Yeah, and that's where we're going to go. Okay. So if you go down here in your phylogeny browser,
I'm still not seeing the same thing as you. That's weird.
Okay. Did you click on genomes up here? That thing?
I see something that says USC genome browser on human January 2022, telomere to telomere, CHM13 version 2, human S1, HS1.
Okay. In the upper right, you've got home. Okay.
Yeah.
I just want you to click on genomes. Just click on that.
Okay.
And you should see this.
Okay. Now I do see that. Okay.
Okay. Cool. Okay. Now I'll go down here and you can pick your animal. And we're gonna pick the human animal right here. You see where I'm at?
Okay. That's super cool.
Okay, pick the human animal. And then it'll go up here and it'll show you the different assemblies. And the one it defaults to is HG38 because that's the one that is most annotated, but it's not the most perfect one. Okay.
Yeah.
This one up here from 2022, that one's from 2013. This one is 2022 and it's HS1.
Yeah.
So pick that one.
Yep.
Okay.
Click.
And then down here in symbol search, we could go CHR2. You see where I'm at? Typing CHR2.
CHR2. Does it matter is it like case sensitive?
No.
Okay.
I believe it is. And then you can go colon and then 1 dash. I'm going to go 1 to 1000. Okay. So I'm looking at the very beginning of the chromosome.
Okay.
CHR2. Okay.
And then hit go.
Yeah, you have to hit go. Return doesn't work on this, so you have to hit go, and then it'll go ahead and it'll show you that. So it's showing you this particular set of bases, right here. Why? Okay, let me try to make this
No
Smaller?
results.
What?
I may have typed the wrong
Oh,
thing.
you might have typed the wrong thing. Okay, let's go back. Oh shit. Okay, go back and on position search term, click on that,
Yeah.
and you have to. For some reason, it has a default in there. You type in CHR2,
Yeah, I had that.
okay, colon.
Oh, yeah, I used a comma instead of a colon. I think you might
Yeah,
have said
comma,
comma previously.
no worky. Okay, we'll do 1 to. Let's do 1 to 300. Okay,
Okay.
1 dash 300.
Yeah.
Okay,
Yeah.
and then hit go, and you should end up at the same place. That's
Yeah, here we
one.
go. Okay, I see a big purple bar.
Yep. Yeah. Let's see. What do I have here? What is that? CHM13. Okay. Click to alter the density. It's got a whole bunch of stuff, but it's going to show you your genes, any genes that you have down here. It'll show, and I've got it set to show simple repeats, which will show transposons. But let's get used to this thing first. So like when you move up here, you can highlight an area.
Hold on. I think I still didn't quite do something right because I see you have a purple bar and a red bar and several other bars. All I've
It
got
could
is
be
this one purple bar. So
okay. It could
let me
be
go back
because
real quick.
it
Did
could
I?
be because I have some tracks selected that you don't have selected. So let's try to get
Oh,
that right.
okay.
Okay.
So we'll go there. And then what are the tracks?
Okay, if you go down here to the tracks, this is
Yes.
where you get to select
Mapping
all
and
the different things you
sequencing,
want.
genes and gene prediction. Yeah.
Right. And I've got
Okay. Everything I have is hidden pretty much, except dense
right.
backpack, base position, sensat annotation, CHM13 unique, and rDNA models. Okay. So
Okay, I've got Sensat. Okay, that's a centromere hidden. Okay.
oh,
Uh
you've got that hidden.
I'm gonna go ahead and hit that as dense. So you have a dense. Okay, microsatellites. We can make that hide that. Microsatellites.
Microsatellites is hidden.
Okay. CHM13 unique. The hell, I don't know what that is. Hide that.
Okay.
Okay. Okay. And so everything else is hidden except for a human liftover.
Okay,
You
you
want
got
that?
rDNA models hidden too. Cat
Yep.
liftoff genes is hidden. NCBI ref sequence is hidden. And then
Yep.
what were the ones that you said were unhidden? Human liftover?
Human liftover on the right here. It's okay. It's on the right
So
side.
hit that as full.
I got it as dense.
Dense. Okay.
You can alter it by clicking on it, but I'll show you that in a minute.
Yeah.
Okay, so we got 3 things on that line, right? Okay, and then down here on genes
Wait.
and
The
gene.
only things you don't have hidden are base position and human leftover, and those are both dense. Did I have that right?
Sensat notation. I've got Sensat annotation set as dense
Okay. I didn't
because
have
that'll
that.
show us. That should show us the centromere, I believe.
But then everything else is hidden. Just
Yep.
those 3. Okay.
Yep.
Yeah, I still
And
just
down
got all I have is a purple bar. Do you need to like refresh? Oh yeah, I see there's a refresh button. Okay.
there's a refresh, but there's more down here. I've got more.
Oh, now I don't even have the purple bar.
Okay, that's good.
Now it's
Okay.
just an empty. There's no colored bars. There's nothing.
Okay, go down to genes and gene predictions.
Okay. Oh, you were just talking about the first box there. Okay. Yeah. Everything is hidden in there.
Yep.
That's all hidden.
And genes and gene predictions.
Yeah.
You want the NCBI refseq. You want to make that dense. Make it dense.
So is
NCBI.
do these terms refer to like how you're assessing the genomes or something? I'm not even sure what the terms really mean.
Yeah, and NCBI refseq is the annotated sequences for all the genes, right?
Okay.
Cat liftoff is something that we don't want to get into right now, but shows you some more genes. So that'll show
Okay.
you some more genes. Okay, on genes and gene predictions, I have cat liftoff set to full. I got NCBI. Well, let's set that to dense too, just to see what happens here. Okay, so I got two things alive: cat liftoff and NCBI RefSeq.
And those are both dense. Okay.
Yep, CRISPR I got shut off.
I'll
Okay.
hit the refresh again.
Okay.
Okay. I'm seeing more categories appearing, but I'm not.
Yep.
There's just no chart. There's no data for the categories.
Yeah.
I'm confused by
Yep.
that.
That's because, well, I'll show you why that is. Because you're in the telomere. Okay, there are no genes over there.
Oh, okay.
Right, so I got everything else hidden: mRNA, expression regulation, comparative genomics, except for on the bottom here, you see repeat masker, variation and repeats.
Hold on. You're going too fast for me. What variation?
Can you go to your studio and get rid of my name on the bottom?
Okay.
Speed of sound.
I can try. Let's see here.
Where the hell is that? I keep forgetting where that is. Let's see.
Edit name. You want me to
No, it's somewhere in StreamYard on the right. You'll have a little panel that tells you what the hell is it, guys? Does somebody know? Banner. Yeah, banner. Where is banner? Top dog. He's not responding yet. Okay, you want to shut banners off. Oh, okay. Well, that's better. You got AIA. You still, yeah. Well, it's still covering the.
Oh,
well, no, it's.
Okay. I see. I did not understand. Okay, my bad.
You see this right here? Repeat masker. We want that on. We want that full.
Okay.
Okay. That's going to be your ERVs and your lines and signs and transposons. Okay.
All right. Hitting the refresh again.
Okay. And then you should be all ready, and we should match at this point if there's a refresh on the bottom. So let's see. I'm going to refresh. And okay. That's what I got. So I got repeat masker. Okay.
That looks pretty much the same as what I got. 2 red lines, sensat annotations, 3 blue lines, RefSeq all, RefSeq curated, RefSeq predicted, RefSeq others. Okay.
Yep. You see that bottom thing? Now, if you put the little hand, you put the little finger over on that simple repeat down on the bottom.
Yeah.
You see what it says? TACCC.
Yeah.
That's a telomere.
Oh, okay.
Yeah. So now what we're going to do is we're going to go up here. You see this zoom in, zoom out?
Yeah.
Okay. We're going to zoom in all the way to base. So you're going to say base.
Okay.
You see where I'm clicking right there?
Yeah, I just clicked it.
Okay. Now you can see them.
Oh, wow.
Those
CTAA, CCC, TAA, CCC. This
telomeres.
is the human genome.
Yep.
We're looking at here.
Yeah. And now if you click with that finger there and slide it over, you can look at the next part. Okay.
Click. Okay.
And it's pretty much a pretty damn good telomere, TAACCC. Except right here, you see it's already degenerate. You see right here, TACC. It's missing an A right there.
What line are you at there? 145?
Yeah, well, 215 about.
Okay, hold on. Let me.
Yeah. I kind of slid over until 215 was on the left.
Oh, I have mine limited. It's not letting me go over past 200.
What? It won't slide over. Like if you grab it, like you click and hold
Oh,
and
there we
then
go. There we go. Yeah.
slide it.
Okay. Ah,
Yeah.
I see.
Okay.
215. And so the line lines up with 4 cytosines, then a thymine.
Let's see. Where did I find? I thought I found an error, but yeah. We got thymine, adenosine, cytosine, and thymine again. It's TACCCT, that sort of thing. But I
Yeah.
thought I found an error over here. I found something. Well, let's see. I'm going to slide over
Oh, I
again.
see a TAACCC, and then TAACCC, and then TAACCCC. There's an extra C here. It
Okay,
goes
okay,
from
yeah,
having 3 C's every time to 4 C's one time
right,
by 215.
yeah. And as you go further in to the chromosome, like I'm at 370.
Here,
Uh,
let's make you the solo so we can see your screen
okay,
what you're doing here.
okay. So I'm over there, and what I'm gonna do is I'm gonna let's see. I've never actually. Okay, I'm gonna move to the right one, and we're looking at CC. Okay, to the right again. We're getting out there. Anyway, the best thing to do here is to zoom out again. So I'm going to zoom out 10 times up here, right?
Okay. Yeah.
Again, now I'm going to slide over a bunch and go see what happens to our telomeres. I'm going to go to 14. I think I'm trying to figure out exactly. Okay, now I'm going to go to base again. Okay, now you see where the telomeres are getting all fucked up.
Is there a way to move to line 1580 faster? Because it's taking me a really long time to get
Oh
there.
yeah. You could go to let's see,
Oh, you can
CH
type it in. Okay, cool.
right CHR2 colon 1555 dash 1600. Say okay. You see
Yeah.
that?
Yep.
Okay. And now you can see where our telomeres, TAACCC, that's a good one, TAA, and we got too many C's. We got like 5 C's now. We got
Wow.
4
Okay.
C's.
Yeah.
Yeah, so
I'm
as you
counting
go in,
5.
yeah. So when
1,
they
2,
ask
3.
when they ask why the telomeres at the fusion site are so degenerate, this is why. Because they are that way in the original chromosomes. Telomeres are not perfect. They have all kinds of errors in them. They're
Interesting.
just they're perfect enough to work.
Yeah.
Yeah, it's a consensus sequence. Anyway, so there I got it. So now we can go up here to zoom out. You want to zoom out like 100 times.
Yeah.
Okay, 100 times. Now you'll see all kinds of genes, right? You see this one right here. Click to let's see. Click to alter
Hold on. I think I'm
density.
not zoomed out the appropriate amount. You said 100 times?
I did it like
Oh.
twice. Yeah,
Oh,
two or
okay.
three. I did
Yeah.
it 2 or 3 times.
Now I'm seeing a bit. Oh my gosh. I'm not seeing the same thing as you, but I am seeing a lot of data here. My
Yeah.
goodness.
So, oh, hang on
It
a
looks
second.
like a lineage.
Yeah, there's a lot of genes here basically that we're looking at. Click to alter. Okay, so if you click on one of these tracks, it'll expand it and make it a little more readable. Okay, so here we have a gene SH3 YL1 to 11, and you can see that there's a whole bunch of different annotations because they've gone in the lab or they've used a computer to decide where these exons are and introns and all that stuff. And they come up with a lot of different results. You can see how many different versions of these genes there are, right? We got alcohol, yeah. But okay, let's
Yeah,
pick one here.
I'm not zoomed in or zoomed out at the right level, so I'm seeing completely different list of genes. I'm looking for the one you said, but I didn't see it.
Okay.
Is there a way to tell what level of zoomed out I am? Like,
What you can do is go to CHR2:1. Are you with me so far?
CHR colon 1.
Dash
Wait, is it the colon comes after CHR1, right?
CHR2:.
CHR2 colon.
Yep. And then 1-460,000.
Okay, bam. All right. And then you said which gene in this list here we're looking at?
Okay, so do you see SH3? Oh, wait.
SH3YL1 to 11, SH3YL1 to 05. Yeah.
Yeah. Okay, do you have that cat lift off? Does yours look like that?
Cat lift off. Where is the cat lift off?
It should be right here, right where my cursor is.
I'm looking. I am looking. I see two big, long olive green bars, but no cat liftoff. I'm wondering if I didn't click the right thing on my filters. Hold on. Let me go down here to the cat liftoff filters. I have cat liftoff genes set to pack.
Okay, so I'm at cat liftoff dense.
Okay, let me refresh and change that and see if that fixes things.
Okay.
Yep, okay, now I see what you see.
Okay. Okay, we're looking at a pretty big section right now. So,
Yeah.
Okay, here's cat. Now, if you click on that, just click on it, it'll expand it. So click on that little squishy part.
The little squishy part. I'm seeing the SH3YL list again. Is that where I'm supposed to be?
Do you see that? I'm not sure. Do you see? Okay. Do you have the dense one here? Is yours looking dense like that?
I see a little chunk of green and a little chunk of blue. I don't think I'm in the right thing though. What?
I'm not sure. If I click on that, maybe you're looking at this. Is it like that?
Let me see here. I might be able to help solve this problem somewhat.
I wish I could see your screen.
Yeah, that's what I was just about to do. Just about to share my screen. Give me 1 second here.
Cat liftoff and NCBR. Let me see here.
All right.
Top dog has to go. Okay, family's good, Top Dog. Go see him. I'm going to. Okay, there you are. Okay, so you've got. Let me see here. You've got cat liftoff. Okay, so if you go up here, like Okay, so I'm going to try to match what you got. And you've got AC. Yep. So I got the same thing you got right now. Okay, you can go back to mine. Be back on mine? Or okay. Okay. Yeah. I think mine is blown up a little more than yours or whatever, but you're looking at the same thing, right? ACO 79779. Austin, are you with me? Can't hear you for some reason. Oh, I lost Austin. I lost you.
Hold on, can you hear me now?
Now you're back. Oh, you're back twice. You're on here twice.
Just using my phone and my laptop to make
Okay.
the stream work a little smoother. But
Okay.
yeah, let me put you back on phone.
Okay. So we've got a bunch of genes here that we're looking at, right? A bunch of different versions of the same gene. Like there's two things that could happen. Number one, the laboratories or the computer models could get it wrong. Number two, it could be that there are different ways that the gene is transcribed. But anyway, I'm looking at this right here, I'm looking at 205, okay?
Yeah.
And what I want to do is I want to go up here and carefully zoom out 3 times with that SH3 YL1 in the middle.
Okay, do you have to like click on it or something?
Yep, you have to click on 3 times.
SH3YL, I just clicked on it once.
Oh, what? No, shit, I zoomed. I meant zoom in, not zoom out. Okay, what did I just do?
Cat lift off genes item details. Gene name SH. Yeah, I think I clicked on the wrong thing. Okay. Oh, go. Yes, right. We're zooming out, right?
Right.
Or zooming in.
Zoom. Yeah, we want to zoom in. So we got that right in the middle. Now we want to zoom in 3 times and see if we can make that bigger. Okay, now you can see the genes a little bigger. Okay.
Hold on. Let me see here. SHYL, SH3YL. Yeah, okay, yeah. Still all the same genes, but yeah, okay, they're bigger now, yeah.
Okay, and this part on the left is it's exon 9 of 9. Okay, here's the tricky part. You see these arrows? That means it reads the reverse strand. It's not reading this strand.
Oh.
It's reading the reverse. Okay.
Oh, interesting. Okay.
So that's going to make our life difficult unless we go way down here to this thing where all these colors are.
Yes.
See the colors?
Pretty colors.
And hit, you see the reverse right there?
Oh, wow. Okay, cool. Yes, that does make things a lot easier. So you hit reverse, and now you're looking at it from the opposite direction.
Right. Yeah, we're looking at the other strand.
Okay, interesting.
Yeah. And I want to, let's see, I want to slide that over a little bit so it's in the middle again, kind of. Okay. And I'm looking at, let's see, this would be the end of the, okay, each of these little vertical bars are exons. So we're going to want to zoom in another 3 times on this. And I don't think that'll hurt us. Okay, yeah, that hurt us a lot.
Oh, wow.
Yeah, I want to zoom back out. Zoom back out 1.5. Okay, now we can see the whole gene.
Okay.
Let's see, which one am I looking at here? I'm looking at 207. Okay.
I am still seeing roughly the same thing as you. Okay, good.
Yeah, okay. I like this one right here. Now, if you click on this, you got the little finger, and you click on that, it's going to tell you about the gene.
Yeah, okay. I was just seeing that with the previous one I accidentally clicked on. SH3YL1-201 is the item.
Okay, you got 201. Yeah, I've got 205. You got 201? Hang on a second. Let's get in the same spot here. 201. Okay, 201's right there. That's the green one. Okay.
Yeah.
On mine. Okay. Oh no, what the hell? I got 213. What did I click? 201.
Yeah, it's the blue one
213.
on mine.
Uh,
Oh, I can see it
really?
on yours too. Go up to your right a
Oh,
little bit, up and right a little bit.
up and right. Let's see.
There you go.
Where's
You
201?
were just next to it. Go back.
Oh, there it is. Okay,
Yeah,
SH1 201. Okay, got it.
yeah.
Okay. Now that tells you on chromosome 2 that it's from 218,399. It goes to 264,287. Okay.
Wow.
Okay.
That does that mean this gene is like 50,000 bits long?
Yep. Yep.
Oh
Yeah.
my
That's because.
goodness.
Do you know why?
It's a
Because of
complex one?
junk DNA.
Oh, it's a junk one.
Well, no, not the
Oh,
whole gene.
there's a lot of junk
A lot
exons
of
and stuff in it.
intron. A lot of intron.
Introns. Right,
Okay.
yeah.
Okay. Now, if you scroll down on that list, you'll see translated protein, predicted mRNA, and genomic sequence. Okay.
Hold up. Okay, yeah, they're links. Translated protein, predicted mRNA, genomic sequence. Yeah.
Click on translated protein, and you'll see that it's methionine, arginine, proline, I think. Isoleucine. Okay. You see the sequence up here in the upper left?
What?
That shows you the amino acids.
That's not just random gibberish. That's information. I thought, okay.
Oh yeah,
I
that's
thought that was just
information
a chunk of letters like a 404 error or something. That's actually, those are all abbreviations for proteins,
for amino acids. Yeah.
amino acids. Okay,
Yep.
cool.
And let's see,
And
proline.
you've got all these pretty much memorized there, huh?
Yeah, but I fuck them up. V is valine, E is glutamic acid, and yeah,
Did
you'll
they
get there.
did they choose to? Is it one letter per, or is it like a 3-letter combination type deal?
It's 1 letter per with 1 letter per amino acid. Okay,
Jeez,
let me go back there.
that's a lot of amino acids in just this one.
Well, that's pretty small. That's a small protein. That's tiny. Let's see here. What do we got?
Oh, it tells you how many it is. There's 342.
Yeah, 342. That's a pretty small protein. So,
This is so fascinating. Okay, this is very cool.
yep. So now SH3YL1 will go out here. SH3YL1, and we'll see what that sucker is. Okay. Gene cards. Okay, it'll give you a whole bunch of shit. What'll tell you about it, and it'll give you a lot of pictures.
Okay,
And
so this chart just gives you the names of all the data, and then you have to go look the stuff yourself up to see what.
yeah, there's usually it has. Let's see. Usually it has. Okay, where's 201 again? Right there.
Excuse me.
Okay. Usually it has a description here that tells you what it does, but this doesn't particularly have one, I think. Okay, so anyway, we're digging into. Oh yeah, right here. Okay, here we go. No, it doesn't. No, it doesn't tell you shit about it. But we're digging into the gene right now. If we wanted to look at just the mRNA part, we could click on the
I'm
middle
having
one.
trouble getting back to that table. It's weird.
Oh, you have to. It keeps going back to the browser, and you keep having to click on the thing to get to the table.
What was the one again? SHY 21 201.
201, yeah.
Yeah.
That should put us in the same spot.
Yep, and then predicted mRNA.
We want to go to genomic sequence first. Okay. If you go to that, you get some options. Okay. We can look at introns, exons, all that shit. And right here, we want to click this top one on the bottom. You see where I am? Exons in uppercase, everything else in lowercase.
Yeah.
Okay. Just like that. Okay. You got it?
Click.
And it hits submit.
Okay, one sec. No, no, go back. I wasn't quite the same as you there.
Oh, I wonder if I'm gonna. Okay, okay. Exons in uppercase.
5 UTR, CDS, 3
3 UTR
introns
introns.
downstream. Oh, okay. I was the same as you. All right.
Yeah, don't click downstream. Don't you don't want that.
And then submit.
One FASTA record per region and then submit. Okay.
Huh.
Now we're looking at
My computer thinks this page is Somali.
Oh, does it?
Yeah.
Okay, so
That translate page from Somali.
at the very top here, we see the first exon, and that's in uppercase exon, right?
Yeah.
And we're going to want to search for.
It's a bunch of cytosines and thiamines, and then a string of guanines. Yeah. And then shortly after it, an AGCA, it switches to, I'm assuming the lower cases are introns, like you said. And then it's all introns for a good long while there.
Yeah. And we can't find any start codon, which is ATG for methionine. That's a start codon.
Yeah.
So that's a 3' untranslated.
This is fascinating. You can literally just use the find function. Is that what you're doing there? Is that why you got a bunch of seconds
I'm using
highlighted? That's so
the browser
cool.
find.
I love that.
Okay. And then I scroll down all the way down to, you see this intron? Okay, I'm going to tell you.
Hold on. No. Tell me what you were searching for with the find function again. Sorry. I was geeking out about how you can do that. What did you put into the find function?
ATG right there. You can see it right here. I just used the browser find for ATG.
And which one are you on out of 797?
I'm on one so far.
Okay. Yeah,
one's right here.
Yeah,
we don't want to worry about that. We want to make that go back up here. And now, okay, we got this huge intron. We got just a little bit of exon. That's the important part. That's the part that makes you alive. The intron is just a whole bunch of probably junk. This is all junk DNA until you get down here. So scroll down to the second exon. Did you find it?
Not yet. What? It's all just a big chunk of text for me. I'm not seeing.
Down there.
I scrolled really. Oh goodness. Okay.
You can search for. Well, I don't know if we want to do that. Oh, there's a search you can do.
Okay. Yes. At the very bottom, I'm seeing it switches back to capitals again.
At the very bottom, you should be right in the middle here on second exon.
Here, let me share my screen again. 1 sec.
Okay. So _201. What you can do is search for that little arrow, like the wedge, greater than sign, and that'll just go from one segment to the next. We could do that. Let me go back up here. Let me try that. Greater than.
Okay.
Okay, if you go all the way to the top and search for greater than. Okay, so what are you looking at here? I'm not sure where you are. Where are you? Okay. So just search for greater than. So Alt F for your browser search. What you doing there? No one is going to watch this stream. This is going to bore the shit out of our friends. we promise we'll get into some stuff here in a while. Where are you, Austin? Oh, I lost you again. Shit. That's why you're not responding to me. Anyway.
Can you hear me now?
Hey. Yeah, I can now. Now you're back.
I'm sharing my screen with you right now. I'm up at the very top.
Okay,
That's why you couldn't hear me. I was talking to you. Sorry.
Yeah, so hit Command F or whatever it is on your Windows machine.
Yeah, the find function.
And search for just the greater than sign.
Yeah. Yeah, that's
okay.
Yeah, that takes you to
the
very top. Yeah.
Okay, so that's the first one. That's the first exon. Now click next. Click the down arrow.
I only have 1 out of 1.
What?
It says there's only one greater than sign.
Really? How in the hell? So what went wrong?
Yep.
What went wrong? Oh, that's why. Okay. Well, we did something wrong. So let's go back.
Okay.
So hit the back browser back arrow.
Yep. Get genomic sequence near gene.
1 FASTA per record. Okay, you got that checked, and you got introns, you got UTR, you got no promoter, you got exons. What the hell happened? You got the same thing I've got.
And you have it to lowercase or to N?
What? Oh, I've exons in uppercase, everything else in lowercase. So you're 2 lowercase mask repeats. Those are mask repeats. We're not. Those don't matter. I don't think.
Let's see here.
And you hit submit, and it should give you the same thing I got. I don't know why you would get something different. Yeah, I've got. I don't think.
Yeah, everything looks the same to me.
Yeah, so hit submit. Yep.
And it asks again if I want to translate the page from Somali.
Oh my God. What the hell is wrong with?
I still only have 1 greater than sign.
Really?
Yeah.
What the hell? Why would you only have 1?
Do you have 2?
I have a lot of them. I have Alt F greater than. I have 19 of them.
Maybe I'm not getting the whole chunk of data here.
Why is that?
Maybe my computer's too slow.
Maybe it isn't ready yet. Well, let's pretend you are, and we'll just go through and I'll show you. So when I search for the second one, I'll find. So I'm looking for. Let's see here. Where am I at? 2 of 19. Okay, so 3 of 19. I don't know why I had more than that. Let's see. Let me see. Yeah, there were 2 on top. Okay, I'm at the second exon. Okay. So you can see my second exon. So now if I search for Alt F ATG, which is a start codon, look at that. So that's the very beginning of the protein sequence right there, ATG. Okay.
Oh, okay.
Yep. And if we look at, let me see here.
There are other things that start a protein sequence, right? Or is that the only one?
That's there are some exceptions that I don't know much about, but yeah, that's pretty much it. Let's start with a methionine.
Okay.
Almost every gene you look at will start with methionine. So
I think I have 1 second. Let me. What did you say it was? Methionine?
Yep. Methionine. It's an amino acid. Let's see. We didn't write down our protein, did we?
No, I don't think we did.
We should have. We should have done that.
I have a piece of a lab book that I did last semester that says UGA is a stop codon, which signals for the release of the protein.
Yeah, or TGA. Yeah, we're looking at the DNA, so we got T's instead of U's. But,
yeah.
Okay. So we got an ATG and then we have a GAC. Okay. Now, if you pull up this table right here and you look for GAC, that should be
hold on. What is this table you pulled up now? I'm Mike. You gotta slow down for me. You go too fast, Mozart. Calm down.
Okay.
The mere Salieri over here is getting lost.
So go to the Wikipedia. I just put a link to the Wikipedia.
Okay. Here we go.
Yep. And that'll give you the codon table for DNA. Okay.
Here, I can stop sharing my screen now real quick. And then Wikipedia.
So what did I say? We had a GAC after the ATG. So we have an M, we have a D. Oh, there's something else we can do on the browser. I'll show you in a minute, which is a lot better.
Oh, I can't just copy and paste the link you put in the chat here by clicking on it on StreamYard.
Yeah, you can click on YouTube. I always keep the YouTube window open too, so I can see my stream and I can click on the chats. But fucking StreamYard doesn't do links.
That's
very well. Anyway, we don't have to. You don't really have to have this.
I just pulled it up.
Okay.
Hey, Robin Webster. Hey, James. Hey, Pragmatic Crystal. Hey, all the great people I didn't say hello to. Regios. Anybody watching this after it's completed, might be more enjoyable to do that, so you don't have to watch me.
Yeah, you could buzz ahead a little bit, right?
It's not showing your most recent chat in the YouTube chat, so I can't click on it there either. Wow, that's frustrating. Why would it not show it? That's odd.
I don't know. Oh, because do I have a wrench on your channel?
I don't think I ever got around to giving you a wrench. I guess we should go ahead and do that now too.
Yep. Yeah, that's why you can't. Yeah,
you're not going to see throwaway stream here. Let's.
You're not going to see any links.
Let's figure out how to give you a wrench.
You go and go up. You got to be in your YouTube.
Oh, in the YouTube.
Yep, YouTube. Looking at your stream, the live stream you got going right now.
Yep, I sure do.
You click next to my name. There's a little ... We used to call those accordions, I think. I don't know what the ... Anyway,
well, your name isn't in there because the only chat you've sent is hidden from me.
Oh, okay. So hi. I'm gonna hit a hi. Okay. So
there we go.
I should.
All right. Add as moderator. That'll give you a wrench, right?
Okay. Now you should see the link. You see the link?
Hold on. I haven't made you a moderator yet.
Oh, God.
There you go. I just made you a mod.
Okay. Let's try again. How about now?
There we go. Yep. Okay. I see it now.
Got a link.
See it now. Okay. Hallelujah.
Okay. Anyway, we were looking at the exon, right? Which is, I'm going to put this over here. So we're looking at ATG, which is methionine. So if you go here and you go A, U, G right here.
This is wild. Okay, I know this table. It's in the back of that lab book I was talking about earlier. Okay, cool.
So this is A, U or T, and then G, and then it says M, methionine.
Okay, cool. I never knew exactly what it was. I knew how to use it well enough to pass the exam.
Yep.
Okay, that is super freaking cool.
Okay.
So this makes sense of that whole stream of data, this thing I copied down earlier, this string of letters that we were
Yep.
looking at.
Yeah. Now,
Okay, that is so cool.
yeah, let's look at the second one, GAC. Okay, right here, GAC.
Yeah.
And if I get that back again, we'll go GAC. Okay, and that's aspartic acid. You know how you
And,
remember that?
and again, this is only for exons, right? Like interons are just, they
Yep.
don't produce anything.
No, they might do some, they might have some function, but it's for the
Yeah.
most part. I want to explain something about introns. If okay, you see how big the introns are compared to the exons, right? You've
Yes, there's
seen
a
that. Okay.
buttload of them. Yeah.
Okay. So if your nucleus took all of those introns and they were all functional and it saved them all, your nucleus would start to swell up the size of Trump's ankles. Okay. It would, and not only that, but you'd completely run out of nucleic acid, so you couldn't make any proteins, right?
Yeah.
So that is an argument for junk DNA. These things get snipped out, and they form this little laureate, which is kind of a weird structure that's different than regular DNA. And that laureate gets hooked up to something called a I forget what it's called. It's a DB protein, and it degrades that intron, turns it back into nucleic acids over and over again. This
Wow.
happens
So
billions of
it's like
times.
if
Yeah.
your drunk jaw gradually decayed over time and like dripped out the back or something like that. Is kind of what I'm
What?
picturing.
What's that? No, it gets turned back into nucleic acids. It gets
Right,
broken
right, yeah.
down.
Yeah. You take this chunk of stuff you don't need from this project or that chunk you don't need from
Right.
that
Yeah.
other
Okay.
project, and you leave it in
Oh,
this drawer, and it slowly
yeah.
over time dissolves into the components to make new tools or new things that you do need.
You got it. You got it. Okay. Okay.
It's
So
a
now
weird metaphor, but my brain is weird.
it's pretty good. Okay. The second codon GAC turns into aspartic acid, and aspartic acid is ASP, but it's represented by a D, and you remember that by saying not aspartic, but aspartic. So
Yeah,
with a D, yeah, pronounce it with a D, aspartic acid. And
aspartic acid. Yeah,
this one up here, glutamic acid, glutamic acid, you say
yeah,
glutamic acid, and it's an E.
yeah,
Glycine's an interesting or easy one. That's just a G. Anyway,
yeah,
you'll get used to these, but you'll learn those. And what is the other one? Arginine with an R, right?
yeah.
So most of them, like lysine with a K, I don't know where the fuck they got that from. But
It,
lysine is,
it's probably some Latin word. Like that's what happens with
yeah, I don't know. Yeah,
on the
yeah.
periodic table with gold and silver because they're named after the Latin aurum. You know?
Yeah.
Yeah, that's what I'm guessing.
Leucine took the L, so they had so I think they just went one down and said, K. We'll call it a K.
Is there
Anyway,
a
they gave it.
is there a hopefully a useful charts video about this chart I can watch? Maybe. Gosh, that
Maybe.
channel is the best.
Oh, I've got a lot of resources I can show you on amino acids. Yeah, I've got some little charts that I love.
I'll need to
That'll,
bookmark this.
yeah, we'll show
My
you. But
evolution folder.
yep. Okay, so we got GAC, which turns into aspartic acid, which is MD, and then we got CTG. So we go here and we look at C, and we look at T, and then G, and that's a leucine. So that's an L. So we got an MDL, right?
Yeah.
Now let's go back. Let's go back over here. Back. Oh, where am I going back to? Oh, going back. Now we got to pick the protein again because of course it goes to the browser. So okay, did you Did you follow me? I went back, got to the browser,
I
clicked
was
on 201.
no, sorry, one sec.
Okay.
Boop, back. Pushing my poor little $200 laptop to its very limit right now.
Yeah, my Mac is frequently at its limit, and I've got a Mac Pro, but it's a 2019. It's already outdated. Why in the fuck? Why in the fuck? Okay, why am I going back?
All right, gotta click the 201 gene
Oh,
again.
oh shit! I shouldn't have went back. I should have done this.
Item SH3YL1 201.
201, right? And then go down here and go down to translated protein again. Oh, something's wrong. Something's wrong. What the fuck?
I got the
There goes
same
my
thing. The link, the 342 amino acids long protein.
Yeah, but it's not an MDL, it's an MNN. So there goes my demonstration here. I was going to show you how clever I am, but it's not working. So let's do something else. Oh, let's try
You
it
still
another
seem
way.
pretty clever to me because I'm still just barely, getting my sea legs with this browser and what it's actually useful for and how to use it. Because, oh my gosh, it's useful for so many things.
Yeah. So we're going to go here and we're going to zoom in on this exon, the second exon.
SH3YL1201.
Yeah, pretty close. And what we're going to do is go up to the top here. And when you click on the top, what it does is it allows you to highlight an area.
Oh, cool.
Yeah. You have to make sure you don't have that little finger, right? Otherwise, it's going to
Yeah.
move your browser.
Yeah.
You'll highlight an area and then you hit zoom in.
Okay, I see drag and select, zoom in.
Yep.
Oh, babushka. Okay, cool.
Okay, you say I have exon 2 of 9.
Yes.
Okay, so I'm going to click over here and I'm going to do that again and I'm going to zoom in some more.
Oh,
Okay,
whoops.
we're zooming in on an exon.
You're clicking on the spot where there's the big blue box, right? Yeah.
Well, no, I'm going up here to the top, sort of, because that'll turn your cursor back into a pointer.
Yeah.
And I don't want that.
Oh, wow. Okay, I think I zoomed in too far because now my whole screen is solid blue and solid green.
Oh, okay. So you may have the wrong part. Escape. I want to go right here to the beginning of this exon. So you got to try to get back to where you can see the exon and go to the beginning.
Okay, yeah.
Otherwise, it takes you forever to scroll with this little finger.
Right.
It takes forever. Okay. Okay. So now, let's see. Click. Okay. See what I did? Okay, I'm going to show you something. Okay, you go up here to where the ACE, where the genome sequence is.
Yeah.
Are you zoomed in as far as I am?
I'm zoomed in a little too far, so let me zoom out a little bit.
Zoom out and try to go to the beginning of that exon.
Okay.
Exon. Oh,
Getting
shit.
closer.
No, this exon 2. Exon 2. Okay.
Okay, here we go. I think I'm a lot closer now. Exon 2. See exon 3 of 10. Exon 4 of 10.
2 of 10.
Exon 3 of 10. Exon intron 2 of 9. Exon 3 of 10. Go straight to intron 2 of 9. I'm missing something. Okay. You said you're looking for 2 of 10? Do I need to zoom in a little further?
Yep, exon 2 of 10. So you might have to zoom out and find your exons again. Go to the second one.
So I need to zoom out.
Zoom out, yeah, so that you can see your exons. I'll go ahead and zoom out with you. I'm going to zoom out 3 times.
How will I know when I can see the exons?
They're the big blue bars. If you got like, okay, you see it. You see it.
Exon 4 of 13. I see big blue bars and I see big green bars. Like
Okay.
the big green bar right now is telling me it's exon 3 of 10. But
Okay, so are you looking at 201 yet? We're looking at 201 right over here.
yeah, SH3YL1201. Yep.
Okay, you're looking at 2 of 10, you said?
Do I need to hold on? I might have dragged my thing a little too far to the right. Might be the problem. 1 second. Exon 4 of 11.
Yeah, you want to.
Exon 4 of 12. Exon 3 of 10.
Okay, if you're on 201, which is right here, SH3.
Okay. Exon 3 of 10.
Okay, so you want to like swipe it over that way to go back to exon 2. And you may have to zoom out again. I'm not sure where you're at. Wish we could see both our screens at the same time.
Yeah, I
That
can
would be
throw it
cool.
up there again. 1 sec. It's just really pushing my computer to its limits.
Okay.
All right. That's great.
Your guy never showed up.
Yeah, it's all right. Life happens. All right,
Yeah, I
you
hate
can
when
see
that.
my screen now.
Okay.
You'll be able
Okay.
to.
Okay. So
Oh, wait,
click
no.
on that.
No, you can't. Hold on 1 second.
Oh no, I'm still looking at mine.
Yeah. Isn't it? Go back. Come on now.
Cool that you can actually see exons, right?
Yeah.
Isn't it? Yeah.
Right.
Like when the creationists give you some bullshit, what you want to do is pull this up and show them the actual goddamn data. Because when you look at the actual goddamn data, things fall apart for them. Oh, I lost you again, Austin. I don't know if you can hear me or not.
Yeah, I can hear you. It's just trying to figure out how to get this to display.
Well, maybe, yeah. Do you use OBS? Are you using OBS?
No, what is that? I don't even know what that is.
Oh my God, yeah, you need to learn about OBS. Something studio, something. What the hell is OBS? I forgot what it stands for.
Isn't it letting me add that to the stage? That's so weird.
OBS Studio. Be cool if, because with OBS, you see, that's if I go up here and shut that off, you can see my old face. God, I'm old. What the fuck happened? I was just 9 years old last time I checked.
Okay, I'm going to remove myself from stage.
There you are. There you are. Oh, you gotta zoom. You gotta zoom out. If you can hear me.
I can hear you.
Okay. You gotta zoom out.
Like 10 times? What do you think?
I would go 3 times at a time.
Whoops. I clicked 10
times. If you do 10, you get lost. Might work. You might be okay with 10. see the problem. We see the problem side by side.
I think we finally cooked my computer, Mike. It's
Oh no. It died. Well, let's go back.
it's ticking and ticking. It's crunching away.
Make me big again, and maybe I can. Maybe you could take notes if you go and try this and keep messing with it. I mean, I've been doing this.
I'm learning how the system works. At least
it's horrible. It's
funky.
it frustrated the shit out of me. ChatGPT has really helped me because you can ask it, "How do I do this?" and it'll lie to you, and then eventually it'll tell you the truth. But anyway.
Yeah, AI is tricky.
Right. So anyway, what I wanted to show you is that if you go to this exon here and kind of move this over to the middle, right?
Okay, finally crunched up, and I do have something that looks almost like what you've got there.
Okay.
So what exon am I looking for?
I'm looking for on 201, it's 2 of 10. On 2 of 9. Okay, this is 2 of 10.
3 of 9. Exon 3 of 10. Exon 4 of 13.
Okay, you got to scroll. You got to swipe to the right so you can see exon 2. Or zoom out a little more so that you can. You're way to the right of me. You're on exon 3 instead of 2. I don't know where exon 3 is. Let's see. Let me go look. Oh, it's way the hell over there. You got a ways to go. Yeah, too much intron. Too much junk DNA. Junk drawer DNA, we'll call it. Could be useful someday. Oh, I lost you again.
Oh, no, I'm still here. Can you hear me?
Oh, yeah, I can hear you. Okay.
Yeah, you said I'm to the right of you or to the... Oh, okay, here we go.
Yeah, you're way over here somewhere.
Exon 2 of 9. Exon 2 of 10.
Okay, you got it. Okay, exon 2 of 10. Okay, so now what I want to do is go up here and zoom in on the beginning of this exon. Like that. Zoom in. And now I can see the actual stuff. Okay.
Yeah,
see the actual.
Highlight it and then do zoom in, right?
Yep. Yeah, you do that. You go up here to the top till your cursor turns into an arrow, whatever. I don't know if your cursor looks like this. This is my Mac version.
How far am I zooming in here?
Well, I zoomed in like, I don't know. Just right at, I just took the very beginning and then I got the A. You know, I can see the codes. Nucleotide.
Okay, I see a big string of AGC. I got big blocks. Okay. Everything looks pretty similar.
Okay, if we go right up here, we should see, let's see, so TGA should be the first one.
Yep, TGA.
Yeah, right there. Okay.
253,370.
Now put your finger on that little list of nucleotides like mine. You see that?
Yeah.
And click. Okay.
Yeah.
See what happened? Okay, these are, what it does is on 3 different tracks, it starts and it tries to figure out which amino acids we got, right?
Okay.
So that's what it's doing.
That is
so what we're gonna do is scroll over until we see a start codon. Okay, and I'm not seeing one. Keep scrolling over.
I see a green M. Is that the start codon?
Yeah. Okay, where is it?
Yeah, I see it.
Oh,
but
right there. Okay,
it's to the left of what I'm looking at. It's near 253380.
Oh, okay. You mean to the left of the exon? It's not
Yeah.
going to work it's not going to work if it's not in the exon. So
Oh,
yeah, you got to be in the exon
okay.
because that's going to get spliced out. Okay. And I'm finding it at, where am I at here? I'm at 253, 285.
Yep. Okay, I see it. Yep, I'm there too.
Okay.
And then
Yep.
there's another one after that, but it's in the interons at 253, 245.
Right. And look at this. It says MDL, just like we decoded. And when we went and looked at the protein, it had a different protein. So I'm thinking they either made a mistake or there's another start codon way over to the right here that we would have to search for about 10 minutes that has the protein coding sequence. Because this right here, if you go here and go down to protein coding sequence, translated protein. Okay, where the fuck is it? Why is mine taking so long? What? Oh, it's MNN-PIP, which is weird.
Oh, that is
So,
weird.
yeah. So that, yeah, I picked a bad example for teaching. But anyway, so that's the beginning MDL, just like we decoded with our
Yeah.
little decoder tool, our Batman decoder tool.
Yeah.
That was created by Batman, by the way.
Yeah, I'm sure it was. Yeah.
Yeah, this thing. Or
Yeah.
what is
Batman or
that?
Bruce Wayne, one of the two.
Yeah, that thing. Batman decoder tool. Anyway.
Yeah.
Okay. So anyway, so that's how you do that. And then you basically you can read your protein sequence from there and then slide over and you keep saying and you know it started, so this third track is the until you fall off the end here. So SFL should be the last piece of the protein. I'm gonna go out here and see if I can find S. Fuck. Let's see. Okay, I'm gonna zoom out again. Anyway, that pretty much wraps that up. If you learn how to use that much, okay, I'm zooming out. I'm trying to find my third. Okay,
What I'm
here.
interested in is the fact that you can use this to compare humans and other species too.
Oh yeah. There's a compare track down on the bottom, but that's like BLAT and stuff like that. Like, okay,
Oh, so you can
yeah,
turn on this primate chain net thing. What does
yep,
that do? Let's turn that on to dense.
yeah. It shows you how we compare with other primates. Though I don't know a lot about that, so not sure. Let's see. Now, what there's a
Oh,
lot of other things you could do here, like if you
there's a bunch
highlight.
more colorful bars at the bottom now that I have no idea what they mean.
Okay, cool.
Interesting.
Well, look at this. You see these repeat maskers? Okay, I
Okay.
click on that. Let's see. Okay, I should be able to click on it to get more density. Now it's going to show me all kinds of ALUs and LTRs or lines and signs and like that. This one is a line L1 family, limb 4B. These are those jumping genes, those things that Donnie loves to talk about as ERVs. And this is an
Yeah.
ALU, which is a sign, and this is a line, and I think this is an ERV right here. This one, yeah, it's an ERV. So HERV, you know, Donnie's always talking about that shit. And it goes ahead and it shows you the DNA sequence and it shows you how well it matches. You can tell that this is mutated all over the goddamn place. Anyway, these are the
My
jumping.
favorites bar has been transforming as I've been learning more and more. It's gone from being like Netflix and Hulu and Amazon Prime to now I've got Google, Gemini, my university webpage, YouTube,
Yep, yep,
Wikipedia, the taxonomy browser, and now I just added this genome browser. Everything
yep,
is becoming more academic for me all the time. I have a whole
yeah,
evolution bookmark file now that's full of thousands of links, literally.
yeah.
Why did I do this to myself?
I know. I'm gonna zoom in on exon 3, and
I'm back at the genome browser page now, and I'm looking at human, mouse, rat, zebrafish, fruit fly, worm, and yeast. These are
cool.
their popular species here. I
Yep.
can see why too. Interesting.
Let's see.
Sorry.
Yeah.
You're looking at the amino acids still here.
I'm looking at the amino acid, and I'm not sure which one. One of these tracks. This is exon 3. I'm trying to figure out why. Well, fuck it. So there's a problem. There's a disconnect. Normally, when you go to that translated protein, you'll find it'll code perfectly from that start protein, methionine ATG, and you can just read it right off. I mean, it usually works pretty well. But let's see, what else is there that I can show you? Well, there's another crazy thing up here, and that is if I go to view, I can look at the entire DNA sequence for what I'm looking at, right? So it'll just drop it down. DNA sequence, it says, okay, all uppercase, I'm going to get DNA. So, wow, that ain't a lot. Yeah, okay, well, that's the part I was looking at right there. So now I can take this like that.
Oh, that is cool.
Right. I can copy that, and I can
and
go.
You can see where it repeats in the.
Yep. And I can go back over here, and now I go up to Tools, and I go to BLAT, which is BLAST on UCSC, and I can paste that in. Okay. Now it's going to search on human, but I want to look for it on chimp. Okay. So I'm going to look for chimp, and I've got Clint, which is the latest 2018 build. It's a T2T build, and I'm feeling lucky, so I'm going to submit it. And it's on chromosome 2A. Lo and behold, right where it should be if there was a fusion. Now I go back to browser, and I can see where the hell is it here? HS1 DNA. Oh my God, it's a little bit different. So I hang on a second. Let me go back to that.
Oh,
okay.
cool.
It was 100%, but it's also found on chromosome 8, on 13, and well, okay, the 130. You got to look at the span. So that's my entire sequence right here.
Holy cow!
Yeah, if you go to browser, and it turns out they don't have the gene annotated, I don't think. They have HS1 DNA. Okay, yeah, this shows you what I searched on, right? That black bar, I must have named it HS1 DNA. And well, maybe
my mind right now, Mike. Slow down a little bit, because holy cow! So I picked a random one and grabbed some detail, grabbed the chain from it and copied and pasted it. It isn't the same one you pasted, I think, but the one I pasted also comes up in chromosome 2A, and it has a 98.8% identity. It also comes up in chromosome 6 with a 100% identity, chromosome 10 with a 96.2%, chromosome 8 with a. It is no wonder they don't show the data when they talk about the data, because oh my God, this is blowing my mind. There's so many 100% in a row.
And look, we're in the chimp, and down here is SA. Here's the same gene, same exact gene.
Oh my God.
And you can go over here, and instead of clicking browser, if you hit details, okay, it'll show you the exact alignment between the human and the chimp. So here's our search sequence from the human right here, and here's the chimp. And these vertical bars means that it aligns exactly. If there's an X, so this is identical, and it's in exactly the right place to be a chromosome fusion.
Holy crap. And it blows my mind, because that's probably the exact same for the bonobo, because I'm looking at the bonobo right now, and the bonobo and the chimp is super similar. Oh my God.
Yeah.
How do they still exist, Mike? How are there still creation at South
I know.
Park?
I don't know. You know, it's like my daughter asked me, "Daddy, why did God make yaks?" I don't know.
I
I have no idea.
Yeah, it's a mystery.
Anyway, that's some cool stuff. But now
That is great.
I want to just give you a quick overview of what I'm working on. Okay.
Yes. Okay.
So I went to
That was
chromosome.
quite a lengthy detour we went on. That was like an hour and a half detour. Let's get back to the main point again finally.
Okay. So here's a fusion site, and I have chromosome 2A, and I started looking at synteny, right? And to look at synteny, you're looking at how the genes match up. And I don't know if you watched Creation Myths when I was on Creation Myths.
Yeah.
I showed that the genes lined up just exactly. But if you go over to, let me see, let me go to GAP. I got to go back to a different spread, different sheet here. CHR2 gaps. Okay. Now, if you go and look at this thing right here, I've got the human, okay, CTNNA2, right? And there's 5 million characters, I think, between, let me see, is it 5 million? Yeah, about 5 million characters between genes. Ignore that anchor D, that's that's a pseudogene. So CTNA2, it's a sulc1, G1, right? But now if I go over to the chimp and look at their annotation, I see CTN1 and sulc or suc-LG. That's that one is a mitochondrion gene, by the way. CTNAA and NA2 is used in the brain to kind of structure your cells. It's really important. But you can go from chimp to human. So the creationists will say, oh, well, what are all these things? Well, when you dig into them, what they are is annotations, basically, that we have done more seriously on the human than on the chimp. Okay. And so I dug in even further and I discovered something. Let me see if I can go to it. Let's see. Okay. Oh, this actually, what you're seeing there, what you were seeing before, I wrote a program to go and pick just one gene out of, there's hundreds of them here, CTNAA, that are annotated. I just pick one. So it's a little misleading that there are gaps there. If you were to compare these more seriously to the chimp, it would be not quite as gappy as you think. Let's see. Okay, we got pseudogene. Okay, that's my, let me go a little further. Okay. So I did, this is the area here, this purple area.
Yeah.
That's basically the area between those two genes that I started to explore. And what I found out is this is the human part, the black part, and then it skips a huge section in the chimp. The chimp has a big insertion right here, right? There.
Yeah.
And then it goes back to the human and then it lines up again. And I call that Betty. Let's see. Okay, where's Betty? Okay, this is Betty, basically. Betty is like if you look over here on the right, you'll see that it's kind of like
that's dark is matching, and everything where there's a gap, it's yeah.
Okay, I was explaining that last night. And if you go to the very top of this, so there's 5 million characters that I need to account for. Okay, it matches perfectly here, and then Betty starts, and it gets all weird, right? So I started searching for it, and what I found was pretty fucking cool. Now there's a transposon, it's a line, it's called L1PA2, that they think is pretty much dead even before we split with chimps, okay? But every now and then they'll find a cancer where this L1PA2 becomes active and it inserts a transposon into the middle of your genome somewhere, and then it kills you, right?
Yeah.
But I found right here that this area matches up. There's about 4 bases between where it quits matching with human. So this is a chimpanzee insert that happened after we split with the chimp.
Oh.
And I discovered this. So you like when you start looking at the details of the genome, you start discovering things. Anyway, and
That's wild.
but now I don't. Okay, there's some other things here. There's let me see if I can make this bigger. I can make that bigger.
And
There's
I can see how this easily and parsimoniously fits an evolutionary model. But there's
exactly
no way this fits a creationist species fixity model.
right
This is evidence humans, and they
because
think is the quintessential, different species.
yep
So if you don't consider changing into a human change in kind, then nothing is evidence of change in kind, and
yeah.
change in kind obviously isn't necessary for evolution. Wow. No, go on. Sorry, it just kind of
Well,
struck
we can.
me a little bit.
So then, but there's this area here that I don't know why it inserted that. It may have done it as a result of this transposon. It may have drugged that in. But right here, we can see that there was a L1M5. So that's a line. That's a different family. They got inserted here, but it's cut in half. And it's cut in half by a jumping sign, SINE,
Yeah.
small interspersed nucleotide or nuclear element. An ALU got stuck right in the middle of that thing right there. So that also shows you a sequence of events, right? That
Yeah.
we had this thing which should have been together. This jumped in the middle. That jumped in the middle and spread the human
It's
chromosome
like
out.
you can almost think about the genome itself as a crime scene, and there's all
Yes,
these little
exactly.
clues around, like this window here got smashed, and this thing
Right.
got scratched like this, and you can see similar scratches and smashed windows at multiple crime scenes, and kind of start to conclude there might be a common perpetrator, as it were.
Yep.
And in this
Exactly.
case, the common perpetrator is common ancestry and,
Evolution.
natural. Yeah, yeah, exactly. Yeah,
Evolution,
yeah,
motherfucker.
yeah.
Anyway,
Evolution.
okay.
Oh,
So the interesting thing is that this is an intron 13 of 19 of that CT or C, what is it? CTNNA2 gene. Okay. That's
yeah,
where it is. So it's in an intron. And because it's in an intron, we can pretty much conclude that it is not functional unless
yeah,
they find clear evidence of functionality that gets snipped out, thrown away. So it's not functional. So that shows you that evolution happened. Okay.
yeah,
Why? But they'll say, oh no, God put that in there because chimpanzees need hair on their back. And so God had to put that in there. But they're going to have to prove that this shit is functional. They're going to
yeah,
have to prove that every intron that gets snipped out sticks around in the DNA or the nucleus and does something, right?
yeah,
Uh, some of them do. There are some introns that have long, oh my God, LNC RNA, basically that has an effect on regulation of the genome.
yeah,
But you'll find like 1 out of 10,000 that do that, or 1 out of 100,000 that do that.
yeah,
So anyway, uh, so this now there's two levels. Me and Scott were talking about this. There's statistics where you go over the whole chromosome, right?
yeah,
And you do statistics. What I'm doing is going way down the rabbit hole, down to those nucleotides, and finding basically uh, all the evidence, right? So I've done a bunch of shit here. Let's see, where the hell is it here? I've got some other blasts that I did, with that sequence with the gap. And let me see if I show the gene up here somewhere. I
My
don't think I
goodness, you
do.
might have gone a whole order of magnitude beyond just mere statistics, my friend. You have laid out some outright parameters here. Like,
I have, yeah.
if you understand statistics, you understand that's a huge compliment too. I mean, fantastic work on this.
Yep, yep. And here's Albert. Basically, that's another gap. There's Betty and there's Albert. And I started working on that one, and I found an ALU, and I found a line one, and then I went out and downloaded canonical versions of these transposons and did comparisons. The other interesting thing I found out about the transposon I discovered
I love the names
is
you've given these guys. I don't want to interrupt, but I just
Albert.
want to take a second. Albert and Bet. Do these have meanings, or is it just fun, goofy name? Like,
I just get attached to them like you name your cat. You know, I name.
That's great.
Yeah, that sort of thing.
Yeah, let's go
And
back
then
to Albert.
I've got all of this data that I've accumulated. I've got Betty analyze, and I can't remember where it is exactly.
Betty
but anyway,
cool.
I found out that this transposon in the chimp is very recent because it matches more than transposons usually do. It's almost a perfect match. And I forget what the hell that is. I've got so many files up here. LPA1 sequence. Okay, that's the actual sequence for the transposon. I don't know where it is.
Some nice light bathroom reading there. Just
Anyway, what I showed is that it's a very recent transposition, so it's an active genome. Anyway, I'm done. I've had it. Let's see, where are we? So these things this is called Freeform on the Mac, and this is where I keep all of my my data, right, for the gap. And then I've got this gigantic one for creationist nonsense. I got that one too. I've got a whole bunch of shit with creationist nonsense that I've tracked over the years. All kinds of stuff.
This is a great way to stay organized. I have just a giant pile of folders and memes.
I know. Oh shit! My closets are filled with papers that I printed out before I learned to read online. Oh, and I got one called Stinky Cell. That's pretty cool.
Stinky Cell.
Stinky Cell. This is all the whole bunch of cell videos. You know, you can see I'm obsessed with them. You know, when you start putting yarn and like I've got all these dots and I mean, you can see I got you see how obsessed I am with Cell. He was excited. He was happy that I was obsessed with him.
You know what? I'm kind of sad T didn't make it today because I feel like he is Sal's evil twin. He's somebody who actually does protein science, and this is his real field, but he just does technology on the side. Whereas Sal is somebody who's a tech expert pretending to be a bio expert, you know, the exact opposite. And he also ironically says the exact opposite of what the real expert says, which they say, you know, proteins, enzymes, all these things evolved over billions of years, just like everything else. But Sal's like, no, thing complicated, couldn't happen. Yeah.
This is evolution of the heart, basically, and I did. So I have a bunch of papers. Like this is by Bishopric, 2005, Evolution of the Heart from Bacteria to Man. That's one of the papers. And then, and I can't remember what troponin does, but it's a protein. But anyway, so I got a whole bunch of shit like that. But I got a whole bunch of these work that I've done in the past. And somebody, please.
One is very well done. I would really like to see somebody try and debate your work on chromosome 2, especially with you so well prepared and so well organized.
Well, I've done all this work.
Katrina said her paper is being reviewed, so it will be published soon, and we can start talking about it publicly soon. Once it's published.
Her paper doesn't work very well. Nor does Tompkins. So anyway, I just covered the fusion site right now. Now I'm done with the gap. Okay, I did my gap thing. I just picked a random 5 million character sequence in chromosome 2. There's 122 million characters, so I got 241 to go. Right?
Yeah.
And I'll be dead before that happens. But anyway, so now I'm going to move on to the centromere, because as soon as I prove to them that they're full of shit on the fusion site, they would say, oh, but what about the centromere and Anchor 3D 30BL? So I'm going on to that one next, and I'm going to dig into that one. So
I'm looking forward to
it. Yep. Anyway, okay, you can make me small.
Yeah, here we go.
Make me small, and I'll make me will make me alive. Oh my God. Who's that? Anyway.
Yeah. No,
So
it's
this is, yeah, I don't know. What else are you going to do with your retirement, right?
yeah.
Fucking.
I think helping scientists sift and shovel through the giant mountain of data they put together is a very civic thing to do, Mike. And
Oh,
you're really good at it. Like I am continually impressed by the fact that you have never gotten like a PhD level formal training in these subjects, and you're still in here
well, do you
sifting
know why?
through the data the way you are. Yeah, because you study it like a madman on your own. You've shown
No,
me your
because
stack of textbooks.
no, because they pay programmers so damn much that I got diverted,
Oh yeah,
basically. Yeah.
yeah,
Yeah. Well, I dropped out of high school. I went to work at a bakery, got married, had a kid at 17. Then I went to work at Erie Mining Company, a taconite mine. Then I quit that job and never found another one and ended up homeless. Then I ended up being a night watchman at a park. And then I ended up moving down to Minneapolis and getting into semiconductor electronics
yeah,
and ended up going to work at a computer store and became a programmer. And from there, 40 years of just getting paid.
yeah.
And yeah, so
I,
and
I,
then in
I don't
50,
know if people get paid like that in that field so much anymore. At least not at that level. Like I'm working a job that's pretty tech heavy, and I don't get any special amount of pay. It's nice, but it's not like not anything to write home about.
I just noticed not only do I have one named Stinky Cell, I've got one named Smeldova. I've got some, and I got protein orchards here, and I got zinc fingers, and I got all this shit, right? Smeldova.
Stinky smell Dorva, huh?
Yeah. Anyway, oh my God, I forget. I got to combine those two sheets because I didn't realize I had. I'm so obsessed with Cell that I got
Yeah,
two of them.
two of
Right? Fuck, I need help.
them.
Okay. Anyway. Yeah, if you ever want, yeah, you ever want to go through Cell Cordova or videos or some of the stuff that Brett has done,
Yeah,
I've got a ton of research on all of them. So
I would 100% be down to do that because they are two of the ones I've been interacting with more recently. Uh, those two and Katrina, we've continued to have, interactions after the debate. So,
I'd love to talk to Katrina. The thing
yeah,
about chromosome 2 is it's a slam dunk. All you have to do is look at how the genes intermesh. They're exact copies. They're not
yeah,
only, and then when I dug into that, like I went right into the exons and I compared human CTNNA2 to chimp, and I found three mutations. Right.
yeah,
So far, I've only gone halfway through the exons. Those three mutations are synchronous. They they do not change the amino acid, so
yeah,
they don't make a difference, right?
right.
And that's the only difference. So the protein that we're using in humans and chimps is identical.
Yeah.
It's the same damn protein. So
Yeah,
I
no, we
don't
were kind of
understand.
seeing
Yeah,
that when we were combing through the data together a little bit, and that's really convincing when you can comb through data together and you're getting the same answers and you're getting answers
yeah,
that match each other and support the same conclusion. And it's kind of like how different disciplines all support evolution. It was a great video, nevertheless, despite it wasn't what we really planned, but I still really enjoyed it,
yep,
and I think I learned a lot, so I
yeah.
appreciate it. And I'm going to be doing
Okay,
more digging into the genetics myself. So thank you so much, Mike, for
yeah.
stopping
And
by and walking me through
anytime,
that.
anytime, particularly in the morning, that you want me to give you instruction on how to use that thing, hit me up and we'll Zoom. We'll do a Zoom call together, and I
Cool.
can walk you through and anything else that you're interested in or that you're working on. So okay,
That's so awesome. Thank you, Mike. I
okay,
appreciate it.
yeah.
Thank you
So
to
and
everybody
this,
who stopped by to watch. Yeah. Sorry. What were you saying?
yeah. I feel sorry for our viewers. That was a lot of detail.
And
A lot of these people don't give a shit about this
please,
stuff, right?
come on. They love
Anyway,
it. They're all science geeks here. It's
okay.
you know what? It works in our favor because then just the people who are really, really interested in getting into the nitty gritty are here. And instead of, a bunch of creationists hooting and squawking in the comments, we have all good, interested people,
Right.
who are like actually asking good questions, actually listening and learning stuff.
Yep.
So I do see lots of great people in the chat. Tori Swensinger, Spooky Bed Hair, Objective Ethics, Robin. Thank you
Oh
all for
shit,
stopping
spooky.
by. Thanks to Top Dog and James and PC and everybody who watches this after and the live stream. Leave a
Yep.
comment. Let us know what you want us to look up on these genome
Okay,
comparison tools next time.
let's get out of here. Goodbye, cruel
Yeah.
world.
Yep. Bye, everybody.